Yeah, first of all, I want to thank you for the organization of this conference. It's such a great opportunity for us to share our research works on the COVID-19 with all of you. And thank you very much for the nice introduction. Okay, to start, actually, I also want to say thank you to the first speaker summit for the nice background introduction. And actually, based on the New York Times database, the United States has just surpassed 5.8 million coronavirus cases and 180,000 fatal cases by yesterday. I'm not sure about all of you, but when we start our explorations of the COVID-19, we immediately notice that errors and anomalies occur in the data, especially under such a pandemic. For example, here we can observe an anomaly in the death count in New Jersey. And when using this data directly for analysis, these outliers may reduce the forecast accuracy due to the bias in the estimate of the model parameters, and a carryover effect of the outlier may also go on to the prediction. As a result, we think a reliable and accurate database is vital for all of us to conduct our further researches and for governments to make better decisions. So therefore, we start our project by collecting the epidemic data from 4 popular open sources: the New York Times, the COVID-19 Data Repository by Johns Hopkins University, the COVID Tracking Project at Atlantic, and the USAFacts. And since we have multiple sources, in the second step, we compare the similarities and differences among them. Then in the third step, we develop some anomaly and outlier detection techniques in the context of count time series. Meanwhile, we are also curious about the reason for those anomalies, so we conduct further investigations on them. For example, we dig into the reason for this big jump, and it's due to that New Jersey started reporting the probable deaths and add 1854 probable deaths, which could date back to the earlier outbreak. And after the anomaly detection, we explore three types of methods to repair the problematic data. The first one is based on the time structure, and the second one is based on the epidemic models, and the third one is based on our newly developed spatial temporal epidemiologic models. And we use all of these 3 different methods to repair our anomalies, and finally, we can get our epidemic database. Okay. Meanwhile, we also noticed that not only the epidemic data is important, local community factors might also influence and shape the spread of the COVID-19 as well, such as the mobility, demographics, and socioeconomics. We also collect those auxiliary information at the county level, and we combine all of them together with our epidemiologic data. And based on the data, we can conduct some other researches, such as our STEM model, and based on the findings, we can establish a dashboard like this with multiple R-Shiny apps embedded in them, as well as a long-term projection for the next 4 months. So in summary, this is an overview of our project, and my presentation today will focus on the first part, the role of the data science in epidemic data comparing and anomaly detection and repairing. Okay. So let's start with the data collection and comparison. We compared our data sources and list the similarities and differences in this table. Here, 1 means the national level data, 2 represents the state level data, and 3 represents the county level data. From this table, you will notice that Atlantic only provides the national and state level data, while the other 3 sources provide data on all 3 levels. Another point we may be interested in here is whether the source includes the data from the islands, such as Puerto Rico and Virgin Islands. Whether the data has unassigned information is also quite useful when we are trying to match the state level and the county level data together. Probably infected and death cases here indicates whether the dataset includes both the confirmed and the probable cases. So besides of these similarities and differences, we also notice that different sources also follow different standards when they report the data. For example, the Atlantic use deaths where the COVID-19 is listed on the death certificate, while the other three sources use deaths among infected people. Okay, so to assess the difference between the two time series, we define this dissimilarity measure. And using this measure, we can effectively detect the counties and the states with the most discrepancy between each pair of the sources, and is meaningful in the comparison between different locations. So here in this figure, we present the county maps of differences of the death count collected from the three data sources. And the area in dark blue in these figures are detected to be different between the corresponding pair of the two sources. You may also ask why they are different. The specific reason for these dissimilarities vary over locations. For example, here you can see some dark blue areas in the New York State. And these are caused by the different geographic assignments. Specifically, NYT and the JHU, they combine the Kings, the Queens, the Bronx, Richmond, those counties together with the New York City, but the USAFacts does not perform this combination. And these two plots illustrate the state-level time series from different sources. For example, Michigan started to report the probable cases and deaths since April the 5th. And probably due to the correction of those probable cases, Atlantic reported a higher infected and death count in the following 3 months. So in summary, the dissimilarity between the data sources are mostly caused by different geographic rules, non-resident data, prison inmates data, and different efforts in correcting the historical data. So from our point of view, the difference in the reported cases do not indicate the inferiority or the superiority of the source. And no matter which source we are using, we need to be very clear and careful about the processing behind it. Okay, after the comparison, now let's discuss the anomaly detection. So within the epidemic data, we observe 2 major types of anomalies. The first one is the order dependency violation, and the second one is the point anomalies. An example of these 2 issues are illustrated here in this figure. And before conducting any analysis of the epidemic data, we may need to cure those issues. So in this section, those examples are based on the NYT database, but actually all the 4 data sources have similar issues. Originally, the order dependency is widely used in the relational database. And in this project, we just incorporate this concept into the anomaly detection and data repairing process for the cumulative time series. So here, order dependency means that the cumulative time series can never decrease. And meanwhile, a point anomaly or an outlier refers to the situation where there is one day of a sudden increase in the cumulative or like the daily new time series. This can also be considered as a violation of the speed constraint. So we use the speed to test whether we have these kind of anomalies. So the anomalies can be caused by a number of factors, such as the release of a large batch of tests and the change of reporting standard, as we just discussed, whether we include the probable cases or not. And besides of those anomalies, we also have some other interesting findings in the epidemic data. The first one is
you have 5 minutes.
All right, thank you. We observed a 7-day cycle in both the infection and death, and this plot shows an example of the infected cases in the United States. And a 7-day cycle can be considered as a special case of the seasonality, so we can use a variety of tests to test for that. And in this table, we list some p-values of the fragment test of the infected and fatal cases. And you can see for the infected cases, 37 states shows the 7-day cyclical pattern at a nominal level 0.05. And for the fatal cases, the 7-day cycle appears in 39 states. Overall, 32 states shows the 7-day cycle in both the infected and fatal cases. And this cyclical pattern is less evident in the county level. So we performed the same test to all the counties. Among all of them, 703 counties shows the cyclical pattern in the infected cases, while 200 shows the cyclical behavior in the fatal cases. And this stacked column bar plots shows the number of weeks that reaches the weekly maximum of the infected and death count across days of the weeks in different states. And this first figure is based on the infected cases. We can see that for the states such as North Carolina, South Carolina, and Virginia, most of the cases are reported on Sundays. And for the states such as the Pennsylvania, or Tennessee, or Wisconsin, most of the cases actually are reported on Fridays. And the next one is based on the death case, and you can see many states reach their peaks on Tuesdays. And sometimes we may experience a pattern change in the time series. For example, here, the increasing speed of the infections in California is significantly different after June the 10th. And there are many possible reasons for these pattern changes. For example, during this summer, we all know that the cases rise in many states as the government eases the restriction and people try to return to their normal routines. And for those change point detection, we just implement the segmented model to detect it. And this table presents the states in which the change points are detected in the daily new infected cases and fatal cases. Also, we specified the point, the date. Okay, now let's think about how to repair these anomalies. To do so, we focus on the daily new infected and death count, and it could be considered as the count time series by nature. And since they are non-negative integers, so we consider a generalized AR and a generalized R models. And the second repairing strategy is based on the model proposed by Altieri's 2020. And this method assembles the following three models. And based on the idea of SIR model, we also propose this discrete time spatial epidemic model, which combines the susceptible and infectious and remote states together. And we model the conditional mean values of the daily new positive cases, fatal cases, and recovery cases via link function like this. And we use the bivariate spline over triangulations to approximate the spatial varying coefficient function beta zeros and beta ones. And to get these triangulations, we can use multiple software packages such as the MATLAB Dismesh or the R package triangulation. Here we only give you some examples. Okay, using the repairing method, we can fix the order dependence violation. Now let's look at the outliers. The outlier correction procedure requirements is different from the traditional ones, since these outliers typically occur with some specific reason. For example, we have two jumps here in this figure, and these jumps is related to the reported cases in the Texas Department of Criminal Justice based on the news on May 31st. At least 82 of the confirmed cases in this Grimes County is related, and this can be traced back to the beginning of the pandemic. So in this project, we developed an automatic procedure for correcting the historical data. First of all, we implement the data repair method we discussed just now to get a reasonable estimate and obtain its corresponding residue. Next, we manually investigate the cause and the problematic period of that outlier. Then we distribute the residue proportional to the value of the time series within the problematic period. For example, here, based on the algorithm we introduced before, we can correct those outliers sequentially.
GuanNan, you should try to wrap up. Your time
this is my last page. Thank you.
Okay.
Yep. Finally, we integrate the clean epidemic data with other county-level characteristics, and the integrated data are openly available here through this GitHub repository. And based on our STEM model, we can also establish a dashboard, as I said before. So we have the short-term prediction and the long-term prediction, as well as a lot of statistical insights. So if you are interested in our product, please scan this QR code to access it. And finally, I want to thank all my collaborators and thank all the frontliner workers. Thank you.
Thank you very much. All the participants should feel free to email the speakers if you want their information and their slides. The next speaker is Changrong Ji.
Hi, good afternoon, everyone. Can you guys hear me?
Yes.
Okay, great. I'm going to share my content. Can you see the presentation slide screen?
Yes, I can.
Okay, great. So let me see, how do I make this thing small? All right, so we have 15 minutes, but we have 4 topics to cover. So I'm just going to fly through all of them in about 10 minutes and then leave 5 minutes for QA. And then also if, if there's people interested in anything, there's the contact info, it's on the back. So the main title for our presentation is a COVID-19 severe outcome risk prediction. We're using machine learning on massive amount of medical records and social data. We're a team of 8. Now, there are actually multiple research aims. One is the top 2 are more data science, specific problem domain solving. The later 2 are more kind of machine learning techniques. So there are mini papers attached to any 4 of these topics. So again, feel free to click on the presentation link and see these mini papers. So first of all, I'm going to fly through the presentation deck real quick. So we are a non-for-profit applied AI R&D organization operating at the intersection of AI, privacy, blockchain, and digital health. And we've got a whole bunch of interesting projects going on in the case in genomics, data science, health analytics, drug safety, blockchain, and the most active one is the privacy and security, private and secure AI. And there's 20-some people on this not-for-profit organization, but 8 of them, quite a few of them are MD PhDs on this particular COVID-19 project team. So the main aim is COVID-19 severe outcome risk prediction, but we're also going to touch on social determinant of health and risk factors. And then the other 2 are kind of, supporting. So we'll get into that. Now, first, let's talk about data. So we are part of a large research consortium. So we are using about 90 million patients, 7 years of claims history, over 3 billion claims line items. And the attributes are like ICD diagnosis code, CPT procedure codes. So among the 98 million people, there are about 200,000 COVID-19 patients. And then we have about 36 million patients, outpatient EHR records with about 16,000 confirmed COVID patients. And then we've got a hodgepodge of social data, including linkage of social data, such as people behavior predictors with a death registry of 80% of US population and 95,000 COVID patients. So these are all de-identified, but these are all personal level data. And they are donated by claims clearing houses, EHR vendors, and social marketing companies. And here are the attributions. So let's focus on the first aim, the primary aim is machine learning for clinical prognosis. So we are going to create machine learning models to predict a patient's risk of severe clinical outcome if they're infected by COVID-19. And the severe outcome is defined as hospitalization, ICU, intubation, ventilation, ECMO, which is a heart-lung bypass, death. So these personalized risk factors obviously can be used in many ways, can help citizens make informed decisions about their work and life choices in the pandemic, help with the clinical prognosis, help with public health. So I'm not going to go through the usual model, machine learning model development. Now, there is a good bit of a feature engineering going on because as we know, claims data is notoriously messy, high dimensional, and sparse. So we're going to use some techniques and machine learning called embeddings, basically project high dimensional data into low dimensional vector space. So there's a whole bunch of very interesting clinical concept embedding work going on in in machine learning. So this is a way of doing feature engineering. Now then there's a data labeling part where we take the 200,000 also patients and look at their ICD diagnosis code and CPT procedure codes and go through kind of a claims manual to label the data, mark them as hospitalization, ICU, whatever outcome we try to predict. Again, we are about a month and a half into a year-long project, so we have some early finding to show. And our number one goal, immediate goal, is to predict somebody's likelihood of hospitalization if they're infected with COVID-19. This is early discovery. So we pull out the top 20 procedures of COVID-19 patients. Again, not going through the details, but these are actually not very surprising for kind of in line with the literature. Top 20 co-occurring diagnoses with COVID-19. Again, this is not prediction. This is just descriptive analytics. Hypertension and all that stuff. So one of the aims of our project is to validate or invalidate these popular beliefs in the literature. Now, but then the second part is to do machine learning, deep learning, to hopefully uncover previously unknown insights. So this is future work. Oops. So we built a machine learning model using the aforementioned data and the label. For this first round, we are using a baseline model of random forest, about 200,000, 200 trees. The hospitalization results yield a report of precision and recall of about 48% and 87% respectively. So for those of you need a quick refresher. So we right now, because it's a baseline model, we're trying to optimize toward recall, try to reduce the false negatives. So we are catching about 87% of the COVID-19 patients in the data set. Then these are the top 25, top 45 features by their importance in the random forest model. And if you look at them, they are, they're actually pretty in line with what the literature says. We used a good bit of manual feature engineering to create about 100 handcrafted features, and these bubbles to the top, things like zip code, which is kind of proxy for people's location, age, heart conditions, chronic kidney conditions, stuff like that. So they are so far pretty in line. So then the second one we did is on social determinants of health and risk factors. Again, we have about 90 attributes of social data from this company called Analytics IQ of over 34 million patients and 95,000 COVID-19 patients. So we look at the impact of demographics, behavioral risks, and predictors, such as motivators. So we took a step at looking at occupation, looking at the COVID-infected people against the total population within that data set with the same occupation. So the bigger bar here is the blue. So basically, the relative difference between the blue bar and the orange bar is the higher likelihood of somebody's getting a higher exposure. In this case, based on population. Based on occupation. So interestingly, because this data is about 1 week old, and schools are still closed and everything. So we are looking at educators, students.
You have 5 minutes.
Okay, great. Educators, students, medical workers, and physicians, we suspect these people might be of particular interest. So the educators are higher risk. Students are low, but will remain to be seen when the schools are open. So so we go to also do by ethnicity, do by comparing the COVID death with COVID infection within the ethnicity. So long story short, the Hispanic population, more than African American, actually has a higher COVID illness burden. So there's more data there. So there's a whole bunch of future work for these two clinical threats or social clinical threats of work. We're going to improve the model performance, add clinical concept embeddings, add a time series temporal features, add more classification categories, and then also you use electronic health record data in addition to claims records and creating more dashboards. And then as the pandemic progresses, we're going to get into, kind of drug and vaccine effectiveness, safety, and then long-term consequences of COVID-19 after we're over the pandemic. And then we're going to get some deep learning models to churn on these data. So I'm just going to touch on this real quick. The constraint of our environment is these data are contained in a very isolated, secure environment due to extremely high sensitivity for privacy and security. So we won't be able to directly use this model trained on patients' data for making predictions. So instead, we're going to use some advanced techniques in collaborative machine learning using obfuscation, aggregation, and knowledge transfer to train the model. To first train the model using the private data, then use the model as a teacher ensemble to train a student machine learning model, and then release that student machine learning model to public for use. And so there's a lot of data sharing challenges, and we got a platform going, and we have the different ways of analyzing the privacy risk, design and model architecture, and this is the very quick high-level survey of privacy-preserving techniques as of 2020, from the data angle, from the machine learning angle, from a cryptography angle, and from the P2P computing, peer-to-peer decentralized computing angle. So it's very vibrant but emerging field of study. How do you do privacy preservation on machine learning? How do you train and learn from the models, learn from the data that you cannot see? It's a very interesting thing. So anyway, so we have all kinds of measures and stuff like that. So I guess I have a couple of minutes. Any quick questions from the audience?
Thank you very much. I don't see any questions, but you are right. You guys are doing a lot of things, and yeah,
Nandini,
interesting.
there are questions, Nandini, in the Q&A.
Okay.
Oh, okay.
Can you read them, Jun? Because I can't see them. I don't know why.
Okay. So first question, if data sets are contributed, they seem to be data of opportunity. How do you know your results generalized to the population? Or what measures do you take to assure that? And how to be sure your data are cleaned and balanced?
Oh, okay. The data is very imbalanced by nature because COVID-19, especially some of these severe outcome like ECMO, heart lung bypass, it's very rare. So they are imbalanced by nature. So we do a lot of machine learning tricks using upsampling, downsampling, all that sort of techniques to address this not uncommon problem. Now, as for the data, because our data set is very, very big to begin with, right now we are doing a 60% training set, 60-20-20. So 60% of the data is used for training, 20% of the data is used for testing. So, so between the training and testing, that's how we tune the machine learning model. And then the remaining 20% is used for validation. So this 20% is not seen, never seen by the machine learning model. And then we use those test sets to make sure, those validation set to make sure that the machine learning model generalized well, not overfitting or underfitting.
The next question is from Peter Meyer. I'm interested in the occupation differences. Where is the occupation value coming from in the data? Is it in the EHR?
Oh, okay. The occupation data comes from the social study of, so we have the tens of millions of citizens with collected, 90 attributes collected by a by this marketing company called Analytics IQ. So they do their thing and they aggregated, they cross-correlated the occupation data and merge it with a claims data set. So that from the claims, we know who are infected, who are not infected, and then the same people have different occupations. So by joining of these two data sets, and then also cross-join with the death index, we do the calculation there.